Ruby is devoted to bioinformatics. Yesterday, we learned host plus the stripe is zebra. Today, I will talk to you about the biology plus information is bioinformatics. Many people think this is the age of big data, but I think this is the age of data science. We handle many high throughput data. It is called big data too. I think it has two kinds. One kind is, file size is small, but there are many files. The other kind is the file size is large, but there are just few files. In general, the data size of bioinformatics, we usually handle 1 billion records for a subject or a person. Is very normal for us. We think therefore the storage demand is increasing year by year. For example, the storage of big drive reached 1 exabytes 3 years ago, and we believe the data size of sequencing will be beyond 150 exabytes in short time. Maybe we think maybe less than 5 years. And besides the storage, the computing power is very required. Your query should run on the high-performance clusters. The program might require many cores of CPU and a large of memories. The I/O efficiency is should be very high because we need to handle many files, handle many records. Then there is a test on American in 2012. It's a protein simulation executed by Cycle computing system. It used 500 cores of CPU and 58 tetrabytes RAMs, and they use 700 American steel instances to run this test, and it just costs 4,800 per hour. I think it's very fair in this cost. But I would still want to ask, is 10G network enough for I/O? The I/O efficiency need to be watched in bioinformatics. The infinite band system used by top 500 system high-performance cluster, and the bandwidth is good enough. It's about 3.8 gigabytes per second. I think the infinite band system is good if you want to build a HPC cluster. But we need to remember that the data science is about data. So we care about the quality of data. We need to decide the factors we want need to be filtered. We must deal the statistics in case of making wrong evaluation. We have to discuss the other people by good statistics. So interpretation might be it's very important because it changed by different people. We care about how to handle a huge amount of data. We must think the data format it must be easy to parse. We want some good statistics tool or module to use by programmers. We love beautiful verification because we can talk with other people and point the important things on this visualization format. So we also concerned about the profits because we need to form a group, a team to do bioinformatics. So we need to concern the profits. Okay, let's talk about some biology. This is a very famous cover page. It talks about the genetics. And I have a dream. Okay, this is a dream. We could find the factor of height of a person, or could find the factor of a disease. There's a technique word called single nucleotide polymorphism. Common single nucleotide polymorphism can explain a part of portion of the heritability of a human height. We show that 45% of variation can be explained by considering all the single nucleotide polymorphism. So, if you are a sick person, could you find the cure one from a lot of medicines, or put your name in this medicine? So it requires the personalized genomic analysis. Okay, so we talk about the DNA. The DNA is a short name of deoxyribonucleic acid. So DNA is composed by the 4 nucleotides. It's adenine, guanine, cytosine, and thymine. So we call the AGCT. It's from a cell, from a chromosome. Okay. This process is called DNA sequencing. So the biologists will do some experiments to get a book of DNA from a specific sample. Specific sample might be the blood sample. Okay, the DNA book is composed of 4 characters. I talk the A-T-C-G. I will not teach you how to read the DNA book, but I will tell you how to do the right things. I just want just choose the right person, do the right test, and most important one, make the right interpretation. Interpretation is very important for data science, also for bioinformatics. And this is an example you might be interested. And the database of bioinformatics is a lot. So the data mapping between database is very complex, like this network. Okay. And I think people here are very good Rubyists. So I think you should feel this is a piece of cake, right? And the bioinformatics, I will tell you some bioinformatics side for the Rubyists. And this is the NCBI, very famous site for the biologists. And many data you can download from NCBI. And this is the ensemble site. Ensemble site is a system written by Perl. So many data is imported to this system, and we can quickly get the data from by the ensemble API. This is a Nature Biotechnology. It's a journal. You can find the newest article or the news from this journal. And this is another journal called PLOS Computational Biology. There are many famous articles. It's an open journal. And if you have any question, you might be asked on the Stack Overflow. But if you have a biology question, you can ask on the BioStars. And there is a forum called Sick Answers. You can ask question here too. And okay, next, I will show you some Ruby site for bioinformatics if you are a biologist. But I think everyone is Rubyist, but you can see, take a look. Everyone knows the GitHub, so I don't tell you that. And RubyGenes is where we find our tools. And Ruby Toolbox help us find the tools. And there is a website called Biogenes. It is a collection of genes for bioinformatics. And there, the most important Ruby gene is called BioRuby. Many genes is based on this BioRuby genes. The other important basic genes is called SciRuby. It's based on the scientific computing tool written in Linux. Okay. Maybe many people will ask, what programming language is best for bioinformatics beginners? I think we should know each row of tools we use in bioinformatics, so we can answer some question of we can give you some answer for this question. This is a mapping sequence data workflow, and it uses the public domain tools like and file format, same file formats and bin file formats to do the variation calling. And this is a simplified workflow of this one. Okay. And as you can see, we use many tools written by different kinds of language in this workflow. We have some popular foundation tools written by C and C++. And each of them use many important key algorithms for bioinformatics. So if you are a C programmer, and if you are interested in algorithm, maybe you can write tools for us. And this is another pipeline used for RNA sequencing. But besides the C language tools, we need some script language tools. Okay, Perl is a very old language used by bioinformatics in at least previous decade. Okay, biology is BioPerl is a very famous module for the for bioinformatics using Perl. The ensemble is built on the BioPerl. Okay, so the other language some people love Java. Okay, I love Java too but I only love the good parts of Java. And JTK is a tool writing for RNA sequencing. It's written by Java. Okay, there are a system called Taverna. It's a workflow system. It's written by Java. And some foundation system, like the Hadoop, is written by Java. So Java is also important in bioinformatics. R. Statistics love R. They do many. They create many tools by R. If you are interested, Bioconductor is the foundation R module for you can get started. Okay, and you can see many data mining analysis books in this link. And Python, young people love Python. I don't know why in bioinformatics. And there's a well-known system called Galaxy. It's written by Python. Okay, so the Ruby in the bioinformatics. What kind of library you think is important? And I list some of these Ruby genes here. Not very overall, but it's a hint. We use active recall very often. We use Nokogiri often. If you want to combine the C library, you can use the FFI gene. If you need to do some parallel, you can use the parallel gene. Okay, and also BioRuby and SciRuby is very basic. And here is some C binding or wrapper genes. The BioSem tools, BWA, BioFE, BioFaster, they are all handle some many different kinds of data formats. We can fast the data from by these genes. There are another kind of language called D language. Some people use D language to create the SemBen bar. Is this gene is something like the same tool but have more efficiency. If you are because the bioinformatics also need to do some statistics. So how to handle matrix is very important. So M matrix is a good gene for us to use. And for the data parser, there are other kinds. So I list a lot here. Some kind of parser is also for another topic like the protein, not the DNA or RNA. And database API or web API is very important because we need to get data from the database. So many people develop the database API or web API. Here is some famous API genes here. I list here. Okay, and we need statics. So, but people use R to do less statics work. And so here is some genes. This state sample can connect with R. They can run some R modules by Ruby. The visualization is very important. We love SVG. We need to create some graphs. So here's some Ruby genes you can use to create SVG or some graph. There are some tools, but I don't list very much. But you can find or Google it on Google it. But if you are not an analyst, I'm just a programmer. What can I do? How can I get involved? Maybe you can work on the pipeline, but there are a pipeline called Galaxy or Taverna, but there are not any open source file system written by Ruby. So maybe you can create one. Okay, or you can do you can create some web system about the data warehousing or some pipeline management, or you can do a coordination center. They handle the general work for bioinformatics, or you can do some work for visualization. Okay, but if this is a beta cloud age, so the cloud, how to do the cloud, how to do distributed computing or parallel computing is a question because there are not very Ruby genes or very or work on bioinformatics to discuss with these topics. So it's a question. I don't know how to answer it. Okay, I'll do it very quickly. In our team, we do some work by Ruby. We create an ensemble virtual machine. Ensemble is a per system. Okay, we use the Ruby diagram chip to create this virtual machine. And you can create very multi-size or multi-space in one virtual machine. And this recipe helps us to build our custom system. And this is a workflow. If you know the chip, you know this workflow. And this is a web view for the ensemble website. Okay, we also modify the draw and sneak pipeline. It's written by C++ bash per Java Ruby. And not Ruby, we derive from this tool and we call it the data row. We add some Ruby scripts on it and do some modification. And it can do the DNI sequence analysis, resequencing analysis. And we put the quality control for DNI. The original pipeline did not do the quality control for RNA. And this is a distributed computing pipeline. So it requires the PBS, LFS, or SG queuing system. The original one only has the PBS. So this is a summary of our pipeline. The green one is the new one we add. The red one is the original one we do some modification. And this is a few web view written by Rails. It's ugly, I know. Okay. And we also do some data parser using Neo4j. Neo4j is we use JRuby to connect with Neo4j. Neo4j is a graph database, so we use it to integrate the clinical research data. We also we handle the Excel CSV file, but this file is collected by different people by different time, in different time. So we think the Neo4j is a good tool to clean up this massive data. And this is a collaboration work between biologists and programmer. And this is a basic algorithm we use to clean up our data. We need an individual one, an individual. They have many kinds of attributes. So we collect data from different kinds of Excel. So each cell have many kinds of records, maybe for individual one or individual two. So this algorithm can quickly clean up the data from different files. And this is a result. Okay, you can see the collection rates of different kinds of data from the different kinds of Excel. The error is about 1.3%. And this is our work. We work for the third-party company. This is API server we write by Rails and run on JRuby. We use Oracle Enhanced Adapter to connect the Oracle database. The third-party company they create a GUI window client for the user, and they will send the XML request to our API server, and we put this API and let this data to the Oracle database. This is a diagram explain how to do this. Okay, this is another example. We create a Rails project and also connect to the Oracle database. User can define a rule for tracking data. Because they usually fill the data in different forms, and we cannot check the form before they fill data. So we need to take a check day by day. This is a very simple overview. So you can see we have a rule editor for user to write how they want to verify their data. This is a dashboard for user to see their rules and how to run it. Okay, and we also do some work for patient randomization. It's also based on JRuby, Rails, and Oracle database. And we need to assign patient to different group. So because we need to choose the right people. So it's by statistics method and randomization method. It's a work cooperate with scientist, programmer and statistician. Okay, this is a very simple overview. People can choose the method for randomization, the size of sample, and the randomization seed, and how they want to how many groups they want to have. Okay, you just put the standard design. This is the results. They tell you each people in which group. Okay, and this is okay, and okay, let's create this one. And database. This is not work for the statistics dashboard. We use the Google Visual Gene for visualization. So we can count some numbers. This is a very simple overview. Okay. So next few things I want to talk. One is about the education. Why education is important? Because bioinformatics is about the biology, about the statistics, about the programming. So the learning is very important. There are two articles tell you about how to learn the bioinformatics. There are some websites teach you how to learn the bioinformatics. But this website mostly use the Python and R. And there's another website. Okay. So I tell you, young people love Python because why? Because there are many books teach them how to do computing by Python. So, and even there is a bioinformatics programming books written by Python. Use Python. So Python is very successful in teaching the real. And so we may wonder, doing like a killer program application written by Ruby, so no people will use Ruby? And this is a question. But as Linda said, we need human. We not need application. So if you are ready, you can please get involved the data science or get involved the bioinformatics. And the next thing I want to talk is about the markets, because we need raise a group. So your product or your work must have some profits to raise your people. This is a study about the bioinformatics markets in global. As you can see, the North American is very high in few years, but the other area is still the same. So it's an underdeveloping market. This study also tell you some topic you can take in action if you want. The data generation and data management, the data analysis and software, data processing storage, and application for bioinformatics for pharma research or developer. And this is a healthcare cloud provided by Dr. Lin Qi-hong in Taiwan. He talked, we can do some work on healthcare in different ways. Okay. The lessons I want to tell you, if you got some interesting, please call for the bioinformatics. Thanks everybody. Thank you.